It's interesting. The more we study the organelles, the more we see that there's quite a bit of interplay. No one organelle is, by itself. They all seem to work together. As we've studied the composition of the different membranes, we've studied how the organelles function, how they repair, replenish, we see that all the membranes seem to come together. They all come from a similar source within the cell. Because of this, we scientists have coined the term the endomembrane system. Endo, meaning within, membrane, the lipid bilayers. So it's a system of membranes within the cell. Come to find out, most of the organelles that take up the space within the cell all seem to have derived from the same source. They are almost completely interchangeable. This endomembrane system, the nucleus, the lysosomes, any of the little vesicles that I've talked about in previous lectures, the endoplasmic reticulum, and the Golgi, all feed into and out of each other. Let's start off with the smallest ones, the lysosomes. Talked about this in a previous lecture. The lysosomes are small membrane bubbles. They are small membrane bubbles that encompass digestive enzymes. These digestive enzymes are not there to damage the cell itself, but are there to get rid of anything the cell comes into contact with. All the cells are voracious eaters in a way, say quote unquote eaters. Anything that comes along that's smaller than them, that is not you, gets engulfed. They call it phagocytosis. The plasma membrane will literally wrap around it. Wrap around it, as we see here. The membrane pushes, wraps around, and it forms a new vesicle. They call this one the food vacuole. The food vacuole will then fuse with a lysosome. The lysosome and all its digestive enzymes will break it down into individual amino acids, into individual sugars, which the cell itself can take what it wants, and whatever else is there that it doesn't want gets pumped out. It's extraneous. It's not needed. The majority of the cell, though, is going to be filled with what is referred to as the endoplasmic reticulum. This is a series of interconnected membrane tubes and sacs. In a previous lecture, I told you, think of these organelles as miniature rooms within a cell. Well, if the ER is a miniature room within a cell, these are all the closets. These are spaces where stuff gets stored, where stuff gets made. The endoplasmic reticulum comes in two flavors. The two flavors, rough and smooth. Rough because, as you see here, the little dots, the ribosomes. This is where proteins are made. Certain proteins, not all proteins. The smooth has a different job. Its job is for the making of lipids, phospholipids, making new membranes. Within these areas, you see here in the light blue on the figure, this is what they call the lumen, the interior. This is where predominantly most of the work is going to be done. And when you look at it, it may look like, oh, this is all by itself, this is all by itself, that's by itself. No, it's just because the cell has been cut in half. And you're looking down in on a cutaway part. All these little areas, this area, that area, this one right here where I drew the line, all of that is linked together in a little maze of pockets. Each pocket, each area has a slightly different pH, has a slightly different composition of what amino acids, what sugars, what enzymes are present. So let's look at the individual portions. The rough ER, also known as RER. And yes, you can use this nomenclature. It is acceptable when you're talking about the endoplasmic reticulum, RER, SER, or just write rough ER, however you want to do it. The ribosomes, once they attach to a messenger RNA, there will be a signal in the before the first few codons. That also includes the first few codons, that as the amino acids are linked together in the peptide bridge, that peptide bond, it signals a halt to the ribosome. The ribosome then moves to the rough ER. It moves to the rough ER, and then once it's there, it will continue making the polypeptide. But instead of making the polypeptide and releasing it into the cytoplasm, it will shove the growing polypeptide into the rough ER. Whatever that polypeptide is going to become, whatever protein it's going to be part of. It is one that the cell does not want free floating around inside of it. So what does it do? Well, it takes that protein, it shoves it into a closet, a portion of the rough ER. Inside there, the enzymes and sugars and other things will push and pull and change the shape of that polypeptide to get it into the correct quaternary shape. Rough ER also makes phospholipids, makes new membrane. New membrane that will break off, as we see right here in this transport vesicle. It'll break off as a new bubble, and within the bubble will be proteins, or embedded in the membrane will be proteins that need to go to the Golgi for final sorting, for final shaping. The smooth ER, as we see here. The smooth ER does not associate with ribosomes. Its little areas, its little closet spaces, its little sacs have other roles. Here, in smooth ER, what we find is synthesis. Synthesis of the carbohydrates, any sugars that a cell is going to make or cell is going to realign, happens in the smooth ER. The making of new lipids, maybe it's the beginning of the phospholipid, maybe it's a cholesterol, maybe it's a triglyceride, maybe it's a steroid. All of that happens there in the smooth ER because it has the correct collection of enzymes to do this. Other enzymes that are going to be housed in a portion of the smooth ER are going to be responsible for detoxification. Detoxification. Turns out a lot of the stuff we eat, drink, that we just don't think about, and doesn't even have to be man-made stuff, just even natural stuff, has within it toxins. I mean, think about it. For those of you listening to this lecture, listen this far, how many of you have eaten an apple? How many of you have accidentally bitten into the apple core and swallowed a seed? Hey, guess what? You just ingested a poison known as cyanide. Granted, it's in small quantities, so it doesn't kill you, but it's there. Yes, the same cyanide you see in all the spy movies where they bite down on the cyanide capsule and it foams in their mouth and kills them within seconds. Yeah, you ate some of that. It didn't damage you, it didn't kill you, because your body transported the cyanide molecules to the smooth ER. Shape dictates function. I've told you this many, many times. And to detoxify something is really not that big a deal. If the protein is shaped like this, this is bad. All you have to have is an enzyme to do this. Get rid of that curlicue, and now you have something that is okay. Twist, tweak, cut, and a paste, and what had the correct shape to be deadly now doesn't. Just undoing that loop at the end. New shape, no function. That's detoxification. The last one I didn't talk about was the storage of calcium. Turns out this is very, very important if you like to have your muscles contract. Turns out that neural impulse travels down the nerve, hits the neuromuscular juncture, jumps that synapse, and what it does is it triggers the release of calcium from specialized areas of the smooth ER known as the sarcoplasmic reticulum. The flooding of calcium within the cell will trigger contraction. Now, in other cells that aren't muscle cells, the release of calcium flooding the cytoplasm will trigger other things. Will trigger the release of hormones that have been stored, will trigger the release of digestive enzymes to fight off an invading bacteria. Many of the things that are going to be made within the endoplasmic reticulum, specifically the proteins made in the rough ER, are then going to be transported from the ER to the Golgi. Why? Well, it turns out the Golgi has completely different internal environments than the ER. Within here, you're going to get the true final quaternary structures of all the proteins. What's important, though, is for many proteins that are going to be involved in cellular metabolism, that are going to be embedded within the plasma membrane to act as transporters or receptors. A lot of them are going to have sugars added to them. We call it sugar moieties. It's not just here's one glucose. No, the glucoses are all linked together, forming specific structures. That can't happen in the ER because that's not the ER's job. Doesn't have the internal environment, doesn't have the enzymes required. The Golgi does. So, as you see here, a vesicle, part of the rough ER, circles around, literally will tear itself off. They call it blebbing, blebs off. and then it'll be moved over to the cis face of the Golgi. The cis face is the face of the Golgi pointed towards the ER. That vesicle, that bubble, will then fuse, become part of the Golgi, releasing its contents inside. As the proteins and whatever move from the different areas back and forth, not only are they going to be individually modified, but they're going to be mixed with other proteins. So where here at step 1, that vesicle may only have a couple copies of a specific protein, all the same. By the time it flows through and gets all the alterations done, its mixing done, and it comes out as, say, a lysosome, it's now going to have hundreds of different proteins present in there. Or comes here to a vesicle that's going to be storage. Again, it's going to be a mix, a mishmash of different proteins that are all needed towards a singular goal. Or the vesicle can travel to the plasma membrane, as I show here. It fuses with, becomes part of, and is doing so, anything inside of it gets disgorged, tossed out. Think of the cells in your salivary gland. You have digestive enzymes in your saliva, your spit. You don't want those free floating around in your salivary cells, the cells of your salivary glands. So precursor is going to be made in the rough ER. It's going to go to the Golgi. Final twist, tweaks, cuts, paste, get it into the correct shape, add any sugars or whatever, going to be put into a vesicle, and then disgorged every time you salivate. At no point in time do those digestive enzymes, that's what these purple things are for our example, these enzymes, at no point in time are they exposed to the cell itself. So the cell is never in danger of being digested from the inside out. Keep in mind, cis face, trans face of the Golgi gives us an orientation, gives us a way of orienting the Golgi as we discuss the different areas within it. Now, for this course, we're not going to discuss the different areas, the different roles and all that. You can do that in a cell biology or a metabolism course, something else. That's an upper division course. The cis, the trans, cis points towards the ER, trans points to the plasma membrane. Gives us an orientation just so that we can know down, up, down, left, right, north, south, same thing there.